Subcommittee will come to order. Chair will recognize himself for an opening statement. Part of the work of our 21st Century Cures initiative is to identify existing roadblocks to speeding treatments and cures to patients. One of these barriers is the current clinical trial process. Among the regulatory and administrative burdens associated with clinical trials are the expanding cost and size. While it takes, on average, approximately 14 years and $2 billion to bring a new drug to the market, a large portion of that cost is spent in recruiting and retaining subjects for clinical trials. It is often difficult to identify potential participants due to a shortage of centralized registries, low awareness of the opportunity to participate in clinical trials, low patient retention, and lack of engagement among community doctors and volunteers. Widespread duplication of effort and cost also occurs because research is fragmented across hundreds of clinical research organizations, sites, and trials, and information regarding both the successes and failures of clinical trials is rarely shared among researchers. Finally, in many cases, researchers have been slow to utilize technology, such as electronic health records and web-based platforms in their trials, which is also a barrier to greater collaboration and information sharing. This expensive and antiquated clinical trials model is simply not acceptable in the 21st century. We can and must do better because patients deserve better. Researchers and physicians are going to have to strengthen the recruitment and retention of volunteers for their trials, adopt new technologies, and above all, collaborate to build efficient and effective clinical trials. I'd like to thank all of our witnesses for being here today. I look forward to hearing their ideas, and I yield the remainder of my time to Dr. Burgess, Vice Chairman of the Subcommittee.
And thank you, Mr. Chairman, for the time, and thanks to our panelists for being here this morning. Certainly look forward to a good and lively discussion. In many ways, randomized clinical trial, this country has set the gold standard for clinical trials, the rigorous investigative approach that we require. It does not mean that you can't make changes, nor that you should not make changes to keep up with emerging science and new techniques in investigational review, all the while keeping a close and careful eye on patient safety. Failure to adapt could see what was once considered to be the standard of excellence in regulation quickly look out of place and out of touch with the field to which it applies. Evidence A, Exhibit A, is personalized medicine and the ability of the human genome to play a role in that. We are approaching a time when treatments could be tailored for a person's specific genetic code. There is no way such a revolutionary approach to treatment could be evaluated in the same way as a single molecule drug meant for large populations. Mr. Chairman, I certainly appreciate the subcommittee asking the question, how can we build in more flexibility? How can we stimulate innovation into the trial process so that these cures, which are just over the horizon, can become the reality of therapies for our patients? These changes must ultimately retain the integrity needed to ensure that the end product is safe and effective. We cannot be caught off guard and risk watching innovative therapies suffocate at the hands of a regulatory system that has not kept up, or further cripple the regulatory system by the approval of products that inherently are unsafe. I welcome the testimony of our witnesses today. I'll yield back to the chairman.
Chair, thanks to the gentleman. Now I recognize the ranking member of the subcommittee, Mr. Pallone, 5 minutes for an opening statement.
Thank you, Chairman Pitts. Today we continue our work on the 21st Century Cures Initiative, and the input from these hearings is valuable to our discussion. One of the primary lessons we've learned thus far, and I expect we'll continue to hear today, is that discovering cures and effective treatments is complicated and difficult. But in the end, when medical advances reach patients, we must ensure that they are safe and effective. And so I welcome today's discussion on clinical trials, which are the foundation of our drug and device regulatory system, as well as the challenges and opportunities there are for modernization of the system. Clinical trials give researchers, drug and device developers, and doctors a way to translate scientific advances into treatments for patients. While not every trial is a success, with every trial, more knowledge is gained about drugs and devices that can be used to aid in the development of a future drug. I think we would all agree that NIH and FDA are world leaders. They have proven that they have the ability and authority to integrate the newest science into their policies and approaches. The NIH-supported Human Genome Project has opened up a world of potential new drug treatment. The groundbreaking public-private collaboration of the Lung Cancer Master Protocol, or LungMap, which we'll hear about from our witnesses today, represents an innovative approach to clinical testing. Meanwhile, just last year, 3/4 of the new drugs approved by FDA were approved in the U.S. before any other country. But there's nothing wrong with always striving to be better. The clinical development phase is the longest and most expensive period of product development, so it's important that we explore new tools, standards, and approaches that can be taken to assess the performance of medical advances. Throughout this initiative, the question remains how Congress can advance these goals. The effort is a worthy one. It's been a great way for members and the public to explore and understand the complexity of issues that goes into discovery, development, and delivery of medicine. But I have to caution my colleagues that when it comes to science, too much or too little is a hard balancing act, especially to dictate in statute. We can't be the science experts. The greatest role Congress can play is ensuring that our federal agencies have the flexibility and resources to apply the best regulatory science available. On Friday, the subcommittee will hold another and related hearing on the engagement of the patient perspective during the development process, and I'm glad that FDA will appear before the subcommittee then to talk about a number of innovative approaches they're taking in their recent regulation of drugs and devices. I think that, Mr. Chairman, I think it's an exciting time in science, and there's some amazing stories to be told, but despite this progress, there is more that can be done. But again, these are complicated issues that I hope we will continue to examine very carefully. I'd like to yield my last 2 minutes to Congresswoman Capps.
Thank you to my colleague for yielding me time, and I thank you, Chairman Pitts and Ranking Member Pallone, for holding this important hearing. I appreciate that this subcommittee wants to take action on this issue. It is a large one. Questions. How do we design a more modern clinical trial? How do we include the right mix of participants so the data are meaningful? How do we ensure that the data analyses performed actually look at differences on gender and ethnicity? How could post-market surveillance and future passive data monitoring help inform our current system? These are just a few of the many critical questions, and I encourage this subcommittee to have additional hearings so that we can truly focus on the many issues under the umbrella of modernizing clinical trials. This is an issue very near and dear to me. For almost 10 years, I have worked to improve clinical trials, and especially those involving women and children. And we have made some progress in recent years, and this has been with the passage of FDASIA and my own National Pediatric Research Network Act. But as you all know, there is much more work to do, and so I thank you all for being here, and I look forward to your testimony, and that's all I have to say. And I could yield back to the ranking member or just yield to any of my colleagues. I'll yield back.
Chair, thanks, gentlelady. Now recognize the chairman of the full committee, Mr. Upton, 5 minutes for an opening statement.
Well, thank you, Mr. Chairman. You know, at our first 21st Century Cures Roundtable, we learned that there are treatments for only about 500 of the more than 7,000 known diseases that affect our nation's patients. We've also heard about the increasing time and expense involved in bringing new drugs and devices to market. And we've learned that the costs and regs surrounding clinical trials are a primary contributor to this delay. This means that new treatments and cures cost more, and they're getting to patients more slowly. That system is simply unsustainable. So here in the U.S., it's incredibly complicated to navigate the processes involved in simply getting a trial up and running, particularly for small companies. Overall, the size, duration, costs, failure rates are higher than ever. In some cases, trials are being moved overseas as a direct result of those challenges. This leaves patients in the U.S. waiting longer for cures and treatments, and it also takes those jobs away from folks here at home. Safety is always the top priority, and I know that we can do better. We must work together to remove any needless administrative or operational burdens that do not benefit patients. In addition, we'd like to learn more about recent advances in technology and data collection that can help modernize our system, encourage better participation, and certainly allow for continued learning about the risks and benefits of new drugs and devices in the real world. How can we take what we learn in the development and delivery phases and translate that back to new innovative discovery in this cycle of cures? How can we leverage patient registries and innovative new protocols like the lung map trial, as well as other collaborative efforts into more advances in molecular medicine? Electronic health records, increased data sharing, and patient-reported outcomes will undoubtedly play a critical role in this regard. Ultimately, it's going to accelerate and modernize the discovery, development, and delivery cycle. So today's hearing is yet another opportunity to discuss what can we do to further our journey on the path to cures. And I yield to Marcia Blackburn.
Thank you, Mr. Chairman, and I want to welcome all of you. We appreciate that you are here as we look at modernizing clinical trials. Federal law requires that medications proposed for human use be safe and efficacious. That means that our constituents can expect medicines to do exactly what they are advertised to do, and that any side effects are going to be clear and apparent to those patients. And the major mechanism by which medicines are found to be safe and efficacious are the phase 3 clinical trials, which test the drugs against placebos and the other known treatments. We all appreciate that process. And what we want to do is look at how we're going to be able to modernize this process as we go through the trials with large groups of people, sometimes thousands, with the intent of finding the side effects that could harm even a small percentage of individuals. The large groups also make the statistics work, giving greater assurance that the drug does do what it is purported to do. The importance of the phase 3 trials is reflected in the statutory language in the FD&C Act. The FDA generally requires drug companies to sponsor at least 2 such clinical trials for a new drug. I would be interested to hear from you. Do you think that's enough? Too much? How should that be changed? Also, the phase 3 trials are the gold standard for drug approval. They have their limitations. How would you address those limitations? Today we're going to look at that gold standard and the limitations of the phase 3 trials and hear of your ways to build upon what we have learned in order to speed safe and efficacious treatments to patients. I thank you for your time, and I yield back to the chairman.
Yield back. Yield back.
Chair, thanks to the gentlelady. Now recognize the ranking member of the full committee, Mr. Waxman. 5 minutes for an opening statement.
Thank you, Mr. Chairman. The topic of this hearing is an important one. Clinical trials are the bedrock of modern medical product development. We rely on clinical trials to demonstrate that our drugs and devices are safe and effective, and we rely on the willingness of people to volunteer to participate in these trials. So, of course, we want to ensure that clinical trials are conducted using the most modern tools and technology that science has to offer. We also need to ensure that clinical trials are conducted in the most efficient manner possible. That's why NIH and FDA have been leaders in working with academia and industry to identify areas in which the clinical trial process can be improved. These improvements could include encouraging the use of centralized institutional review boards, developing standards for harmonizing the collection and exchange of data, and maintenance of patient registries to facilitate the recruitment of patients for clinical trials. And I look forward to hearing more today about such efforts. How Congress can help advance these goals is a complicated question. The 21st Century Cures Initiative is useful because it is shining a light on some important issues surrounding how drugs and devices are developed and ultimately delivered to patients. There are some clear areas where Congress could legislate. We should ensure that both FDA and NIH have the resources they need to remain the gold standard in observing clinical trials. But when it comes to legislating how clinical trials are conducted, we need to proceed with great caution. Congress should not be in the business of dictating the kind or level of evidence needed to permit drugs and devices to go onto the market. That decision is solely the task of the scientific experts at the Food and Drug Administration. We should not force FDA to prematurely accept novel technologies. Our job should be to ensure that FDA has the regulatory authority needed to make use of the latest scientific advances. When FDA testifies on Friday, the agency can tell us about how it is applying novel approaches to clinical trials in their regulation of drugs and devices. I would also like to know whether the agency believes it has the authorities necessary to adopt new approaches and whether other new statutory powers are necessary. In this area, we need to be careful not to try to fix things that are not broken. That could harm a system that is already working. We should create policies that foster scientific advances, but we should not enact regulatory policies based on how far we wish scientific development has progressed. Thank you, Mr. Chairman, and I'm willing to yield my time to anyone who might want it. Otherwise, I yield it back.
Chair, thanks, gentlemen. That concludes the opening oral statements of the members. All members' written opening statements will be made a part of the record. We have one panel today with 7 witnesses, and I'll introduce them in the order that they present their testimony. First, Dr. Robert Meyer, Director, Virginia Center for Translational and Regulatory Sciences, University of Virginia School of Medicine. Dr. Aaron Kasselheim, Assistant Professor of Medicine, Harvard Medical School and Director, Program on Regulation, Therapeutics, and Law Division of Pharmacoepidemiology and Pharmacoeconomics, Brigham and Women's Hospital. And Mr. Bill Murray, President and CEO of Medical Device Innovation Consortium. And Dr. Jay Siegel, Chief Biotechnology Office and Head, Scientific Strategy and Policy, Johnson & Johnson. Dr. Roy Herbst, Chief of Medical Oncology, Yale Cancer Center. Dr. Sandeep Khosla, Director, Center for Clinical and Translational Science, Mayo Clinic. And Ms. Paula Brown Stafford, President, Clinical Development, Quintiles. Thank you for coming. You will each have 5 minutes to summarize your testimony, and your written testimony will be placed in the record. Dr. Meyer, we'll start with you. You're recognized for 5 minutes for an opening statement.
Thank you, Chairman Pitts, Ranking Member Pallone, and members of the committee. As stated, I am Dr. Bob Meyer, where I direct the Center for Translational and Regulatory Sciences at the University of Virginia. I am by background a pulmonary physician and previously held senior leadership roles within the Center for Drug Evaluation and Research at FDA, as well as in Merck Research Labs, where I headed global regulatory strategy, policy, and drug safety, and was a key participant in their late-stage development committee, which was the committee that was responsible for the oversight of late-stage development trials within Merck's portfolio. While I'm now in academics, I think I have a very real and tangible experience with regard to the clinical trials challenges from both a regulatory and industry perspective, and therefore I'm pleased to be here today. The modern clinical development programs are large, complex, and usually global in scope and in conduct, and are increasingly expensive to conduct. Compounding this rising cost is the fact that the success rate for drugs entering into phase 3 to achieve final regulatory approval is falling, and the rate is now approximating only 50%. There are a myriad of drivers that have contributed to the growth in larger, longer, and more complex phase 3 trials, including regulatory demands. However, I think it's important to focus beyond FDA in the considerations on how to address some of these issues. And let me speak to a few of these. I would say that I'm going to keep this statement short because I believe many of these points will be more eloquently made by others on the panel. The first consideration that I'd raise is better trial standardization. In Phase 3 programs, there's a large amount of time expended getting from study concept to the first patient enrolled. And the sponsors usually recapitulate these efforts for each program as if each one is a wholly new effort. This then raises two important points for consideration. The first is the enhanced development of effective, lasting, durable clinical trials networks. Networks can bring efficiencies such as having identified patient populations and qualified and ready clinical sites that can reduce some of the time and effort spent in study startups. There are efforts towards clinical trial network development in certain disease areas, such as the National Cancer Trials Network. However, this model is not as widespread as it should be or could be, particularly taking into account the varied areas of unmet medical needs. Second concept is the development of master protocols. Such master protocols could serve as the basis for use by different investigators or sponsors with minimal modification, save for the details of the particular test product. An added benefit of wider use of shared standardized protocols is this would also enhance the ability to interpret these trials in cross-study comparisons to assess relative efficacy, safety, or other attributes considered important to physicians, patients, and payers, since the patient populations and endpoints would be highly similar. Another consideration is the increasing complexity in design of modern clinical trials. This trend to increasing complexity is reflective of the fact that modern trials are designed to address an increasing number of demands, from differing regulatory demands across the globe, differing payer expectations, differing market claims sought, the use of new exploratory science or endpoints within the trials, and the interest and input of key opinion leaders who participate in the design of the trials. I believe sponsors could benefit from further concerted efforts to simplify trials by using multidisciplinary groups within the company and outside the companies tasked to maximize the value of the trial while minimizing the complexity and cost. I also believe FDA could aid in this effort in the end of Phase 2 advice, but to do so, they would need to recruit more experienced industry personnel with practical clinical trial design and operations experience, because this kind of expertise is rare within the agency. An additional consideration in reducing clinical trial expenditures is moving further away from the paradigm of face-to-face clinical evaluations as the gold standard for patient evaluation. There's an increasingly sophisticated ability to assess patient status and accrue sophisticated clinical data via new technologies. So in light of the other expertise in the panel, let me close by saying these efforts to think about how we can modernize clinical trials are critically important. However, I think that the evaluation of safety and efficacy is a critical safeguard to patients within the U.S., and I think the way that this currently is done within the U.S. is, in fact, the gold standard, not only within the U.S., but across the globe. And I would urge that the increasing daunting costs and the challenges of medical clinical trials are addressed in a way that preserves the assurance that drugs on the market are safe and effective. We must seek a way to deploy into practice the efficient modern clinical trials, incorporate new technology and science where appropriate and validated, while maintaining the integrity of the regulatory process. Thank you for this opportunity to participate in the hearing.
Chair, thanks, gentlemen. Now recognizes Dr. Kesselheim for 5 minutes for an opening statement.
Thanks very much, Subcommittee Chairman Pitts, Ranking Member Pallone, and members. I'm Aaron Kesselheim. I'm a physician, lawyer, and health policy researcher at Harvard Medical School, and it's an honor to have the opportunity to share my thoughts with you about modernizing clinical trials and helping expedite access to new prescription drugs and medical devices. About 50 years ago, Congress decided that new therapeutics should have their efficacy and safety demonstrated before they could be widely used by patients. This wasn't a capricious attempt by legislators to prevent patients from getting the treatments they need, but a rational response by public servants to major public health tragedies caused by the lack of such proof. When Congress originally gave FDA this power, it did not require any particular kind of test. All that's statutorily required is that manufacturers provide substantial evidence that the drug will have the effect it purports to have, with substantial evidence being defined as adequate and well-controlled investigation. Unfortunately, some manufacturers will not subject their healthcare products to studies meeting even these minimal criteria without the FDA's standard-setting authority. Take a look at the dietary supplement market if you don't believe me. Indeed, in the decade after these regulations were put in place, FDA regulators removed hundreds of drugs that failed to show sufficient evidence of effectiveness upon clinical study. To meet these criteria, the FDA prefers randomized trials with blinded assignment in placebo or active comparator controls, and so does the world scientific community. It's worth recalling that a randomized controlled trial was once an innovation. The basic requirements for conducting these trials became recognized and codified slowly over the course of the 20th century after decades of debate and consideration, leading to consensus about their most important characteristics. At the same time, subjecting a new product to a formal randomized controlled trial or testing a hard clinical endpoint could delay availability of promising products to some patients in life-threatening circumstances. Fortunately, as currently written, the law gives the FDA flexibility to accept data short of traditional randomized trials to approve therapeutics for important unmet needs or where randomization may be ethically or practically impossible. These products may get assigned by the FDA to special fast-track or accelerated approval pathways, or receive congressionally authorized designations that signal their special status, like orphan drug, or breakthrough drug, or humanitarian device. Studies conducted by myself and others show that products with these designations are often provided with expedited review by the FDA, many receiving approval based on uncontrolled studies in small populations. Expedited approval pathways and special designations are common at the FDA. In 2012, 26 of the 39 new drugs approved qualified for at least one such program, and the FDA now approves about two-thirds of new drugs earlier than its counterparts in Europe. When medical products are approved without being subject to randomized trials testing real clinical endpoints, it puts patient at increased risk. Medical history is littered with drugs and devices approved on the basis of unvalidated biomarkers that have their indications later withdrawn or altered, or cancer drugs originally approved on an uncontrolled trial, later demonstrated in better controlled trials, finally conducted a decade later to actually increase the risk of death. In 2012, the multi-drug-resistant tuberculosis drug bedaquiline was approved on the basis of 2 short-term trials testing about 200 patients after being granted accelerated approval status, fast-track, orphan drug status, and priority review. In these studies, the drug was only shown to improve the questionable surrogate endpoint of converting sputum from tuberculosis positive to negative, but 2.5 times as many patients died from tuberculosis in the bedaquiline group than in the control group. Patients with tuberculosis want to be cured. They don't want to die with cleaner sputum. How do patients and individual physicians now make sound benefit-risk determinations about this drug or others like it in the absence of more conclusive scientific data? The prospect of approving more drugs on the basis of trial designs that diverge from traditional randomized trials also puts pressure on the timely conduct of confirmatory clinical trials and post-approval surveillance systems. But studies show that manufacturers' commitments to continue studying their products after approval may be delayed or incomplete. Once a drug is FDA approved for a certain indication, convincing patients to subject themselves to further randomized trials of the drug for that indication can be challenging because patients can receive the drug outside the trial. It's no wonder that the FDA gave the makers of bedaquiline until 2022 to complete confirmatory trials of that drug's effectiveness in tuberculosis. In summary, the prospect that researchers can design new ways of conducting clinical trials of investigational drugs is exciting, and I hope that the best of these truncated designs are proven to provide the same level of confidence as standard randomized controlled trials. But the FDA already has the flexibility in its laws and regulations to accept innovative study designs short of randomized trials and validated biomarkers that can accelerate the testing of truly important new drugs and medical devices. The fast-track process reduced clinical development time of a new drug from 8.9 to 6.2 years. Accelerated approval drugs have an average of just 4.2 years of development. And the FDA already exercises this flexibility to a remarkable extent. If regulators and others in the medical community are still skeptical about certain biomarkers and clinical trial designs, it's probably because the science supporting them is still in its infancy, in which case forcing approval of the drugs or devices to which they're applied would be dangerous and counterproductive for the very patients we're all trying to help. Thank you.
Chair thanks the gentleman. Now recognizes Mr. Murray, 5 minutes for an opening statement.
Chairman Pitts, Ranking Member Pallone, and Subcommittee members, thank you for the opportunity to testify. My name is Bill Murray, and I am President and CEO of the Medical Device Innovation Consortium. During my 25 years in this industry, I have had the opportunity to lead multi-billion-dollar global businesses as well as 2 early-stage companies. These innovative businesses were founded on technology developed in the United States. In recent years, however, these businesses have faced a more difficult regulatory and reimbursement environment in the United States, which is challenging our country's position as a global leader in medical device innovation. I applaud the committee's bipartisan leadership in initiating the 21st Century Cures call to action and its commitment to finding solutions to help the U.S. healthcare industry maintain global leadership. MDIC is a public-private partnership between government agencies, including FDA, CMS, and NIH, nonprofits, and industry. MDIC is focused on the medical device ecosystem. We collaborate on advancing regulatory science, by which I mean the tools, standards, and approaches that regulators and innovators use in the development and review of medical devices. We believe that improving regulatory science will offer concrete ways to make patient access to new medical technologies faster, safer, and more cost-effective. Clinical trials are amongst the biggest challenges. The time, complexity, and cost of conducting clinical trials, along with the uncertainty of outcomes, makes them a challenge for both regulators and innovators. And based on a survey of over 200 medical device technology companies, it takes an average of 6.5 years and $36 million before a new Class III device even reaches the pivotal study. We need new approaches if we are to continue fostering innovation. MDIC's goal is to improve the safety and effectiveness of products being introduced to the market, reduce clinical trial timelines and costs, and give U.S. patients earlier access to beneficial technologies. MDIC's work includes several high-priority initiatives. First, MDIC is working to improve the design of clinical trials. Medical device clinical trials are increasingly complicated. MDIC is examining current trial designs to better understand how much of the collected data are used and the ways in which clinical trials may be unnecessarily complex. We are exploring possible alternative trial designs that still supply high-quality data on the safety and effectiveness of medical devices. MDIC is also supportive of FDA's Center for Devices and Radiological Health's efforts to balance pre- and post-market data requirements. Providing a reasonable threshold for clinical data during the pre-market process while continuing to collect data in the post-market setting is a win for patients and innovators. Second, MDIC is investigating ways to reduce the barriers to conducting early feasibility studies in the United States. These first-in-human studies are a critical step in the approval process of many new medical devices, but increasingly they are performed outside the United States. The reasons for this include economic incentives offered by other countries for companies to invest in abroad, but they also include concerns that the regulatory approval process is slower, less predictable, and less flexible in the United States. As a result, U.S. patients often have to wait longer for access to new medical devices. CDRH recognizes this issue and has taken initial steps to address it through a new policy in 2012. MDIC is building on that work by exploring new methods and tools that support each early feasibility studies, such as incorporating validated computational modeling and simulation data into the assessment process. We feel strongly that American patients should be the first to benefit from cutting-edge American technologies. Third, MDIC is conducting research to better understand the data on patient preferences about the benefits and risks of medical devices. Supported by funding from FDA, MDIC is developing a catalog of scientifically valid ways to measure patient perspectives, and we are developing a framework that could support the use of the data in the regulatory process. Fourth, MDIC is convening experts to help the medical device industry harness the power of computational modeling and simulation. Currently, medical devices lag behind such fields as aerospace and automotive in the use of modeling and simulation tools. The development and use of regulatory-grade tools has the potential to revolutionize the field, enabling developers to generate more groundbreaking ideas, test them with greater confidence, and bring them to patients more safely and quickly, while reducing the cost of clinical trials. Moreover, modeling and simulation may soon play a larger role in the treatment planning and the realization of personalized medicine in the clinic. MDIC is making progress on these important initiatives, but more needs to be done. We encourage Congress to support efforts that strengthen regulatory science and facilitate public-private partnership collaborations to improve the innovation environment in the United States. Thank you again for the opportunity to testify about MDIC's collaborative efforts to support medical device innovation that will benefit patients. I will be happy to answer any questions.
Chair, thanks, gentlemen, and now recognize Dr. Siegel, 5 minutes for an opening statement.
Thank you, Chairman Pitts and Ranking Member Pallone and members of the committee. I have been working on clinical trial improvements for over 30 years, from the diverse perspectives of a senior U.S. FDA official.
Is your mic on? Make sure. Thank you.
I have been working on clinical trial improvements for over 30 years from the diverse perspectives of a senior U.S. FDA official, an industry R&D leader at Johnson & Johnson, and a participant in many broad collaborations, including the International Collaboration for Harmonization, the Society for Clinical Trials, and the Clinical Trials Transformation Initiative. I applaud and thank the committee for the 21st Century Cures Initiative and today's focus on clinical trials modernization. Our clinical research enterprise is critically important for medical progress, but was largely designed for conditions that prevailed years or decades ago. We have before us new tools and opportunities to modernize it, and thereby to usher in a new era of efficient translation of scientific advances into medical advances and 21st century cures. I will briefly discuss 4 of these opportunities: use of electronic health records, use of biomarkers, creation and use of clinical trial networks and consortia, and engaging patients as collaborators in the research process. The adoption of electronic health records provides the potential to collect data efficiently in the settings in which healthcare is being delivered, creating a learning healthcare system. Large-scale registries of patients with a shared condition can be constructed, allowing studies of disease course, risk factors, biomarkers, and treatment effects. The powerful tool of randomization could be applied to such cohorts, creating large, simple clinical trials in the care setting. The resultant enhancement of the ability to learn about the effects of medicinal products while in clinical use could allow earlier availability of important new therapies, with assurance that additional information would be collected reliably and efficiently after approval. Full realization of the promise that electronic health record enhanced research holds will require addressing several needs, including standardization, interoperability, and data quality of the systems, research into how best to compile and use the data, and reassessment of the regulatory frameworks that protect patients. The rapidly increasing ability to collect and analyze genomic, proteomic, imaging, and other information allows incorporating that information into clinical trials as biomarkers. One valuable use of biomarkers in clinical trials is as surrogate endpoints, which, if reasonably likely to predict clinical benefit, can support the accelerated approval of new therapies. The success of accelerated approvals in bringing important new drugs to patients in need sooner, together with the ability to measure many new biomarkers, suggests that wider usage of biomarkers for accelerated approval would be beneficial. In the FDA Safety and Innovation Act of 2012, Congress encouraged such wider usage. Use of biomarkers for patient subgrouping and response monitoring can crucially enhance several other aspects of clinical research, including personalized medicine research, disease prevention research, and adaptive clinical trials. Government, in partnership with academia, patient groups, and industry, can create and operate clinical trial networks that provide a rapid and efficient means for assessing promising new therapies. Networks have already led to substantial advances in clinical research, and there's potential to address more diseases, to create broad consortia, and to utilize powerful new tools, such as electronic health record-based trials and ongoing biomarker-driven adaptive design trials, such as LungMap. Patients bring to clinical research valuable perspectives and insights, and often strong motivation to contribute. Enhanced participation of patients in the design and conduct of clinical trials can be expected to improve many aspects of trials. Patient-reported outcomes, together with patient-informed risk-benefit assessments, should play a larger role in clinical trials and product development. Additionally, efforts to involve more patients in clinical research will help unleash the power of a learning healthcare system, while helping ensure that our medical knowledge is derived from the experience of a more diverse and representative population. Mr. Chairman, I thank you and the committee for your invitation and for your attention.
Chair thanks the gentleman. Now recognize Dr. Herbst, 5 minutes for opening statement.
Good morning, Chairman Upton, Ranking Member Waxman, Subcommittee Chairman Pitts, Ranking Member Pallone, and members of the subcommittee. Thank you for inviting me today to share my experience regarding innovative clinical trials for cancer patients. I am Dr. Roy Herbst, and in my role as Chief of Oncology at Yale, I care for patients with lung cancer, conduct and collaborate on basic research, and work on clinical trials from Phase 1, first in human, to Phase 3. Over the last 2 years, I've been working with the Friends of Cancer Research, which was founded and is led by Ellen Siegel, the National Cancer Institute, SWOG, a cancer cooperative group, and the FDA on an innovative public-private partnership approach to clinical trials, and I am honored to be invited to participate in this important hearing today. Cancer is the second most common cause of death in the United States, with over 500,000 Americans expected to die of this disease in 2014. Cancer is a disease that is accompanied by much pain and suffering, loss of life, and productivity. Despite advancements in surgery and drug therapy, many cancers remain incurable. Lung cancer, the #1 cause of cancer death, is one such disease, and as a specialist in this area, I often see patients with advanced disease who have very limited treatment options. For this reason, together with my colleagues in the field, we strive to develop new therapies for these patients so that we may provide them with a cure, or at least with more quality of life and time with their families. We're working hard to personalize care. We want to match a patient's tumor profile with the best treatment, with the overarching goal to find ways to provide more active, less toxic, and more cost-effective therapies. I am happy to say we're making progress. Due to the country's investment in research, in 2014, we can now sequence every gene in a tumor, including the 25,000 protein-coding genes. This is amazing technology and science. However, it remains limited. Why? Because 1, it is still only available to a minority of patients. 2, it is expensive and often not covered by insurance. 3, the informatics and data interpretation challenges are overwhelming. And most importantly, we still do not know how to translate this information into therapeutic benefit. Hence, clinical trials are essential for this process, and the need to modernize for the molecular age is very important. Often, clinical trials are limited by numerous challenges, including the startup time, accrual expense, and the need to identify defined subpopulations of patients that makes trial enrollment difficult. Developing a potential therapy from the initial discovery stage through clinical testing and regulatory approval is a complicated, expensive, and often inefficient process that can take up to 15 years. Let me give you an example. In recent years, we tried to study a drug that affects 10% of patients with lung cancer. That meant we had to screen 100 patients at Yale to find 10. Only 6 of those patients were then eligible with good enough status to go on the trial. We treated 2. That's totally unacceptable. It's not good for the patients. It's not good for the clinical trial. It's not going to advance our cause. With this in mind, the Lung Cancer Master Protocol, known as LungMap, is an innovative, groundbreaking clinical trial designed to facilitate efficiencies and advance the development of targeted therapies for squamous cell lung cancer of the lung, one of the worst types of this cancer. The concept of the LungMap was developed at the 2012 Friends of Cancer Research Brookings Conference on Clinical Cancer Research, and at the same time by the National Cancer Institute Lung Cancer Steering Committee. Since the release of that initial concept paper, through the intense collaboration of many, LungMap was initiated and opened in a very rapid year and a half. The goal is to develop a biologically driven approach building on the NCI-funded Cancer Genome Atlas, TCGA, to identify targets. In LungMap, a master protocol will govern how multiple drugs, each targeting a different biomarker, will be tested as potential treatments for lung cancer. Each arm of the study will test a different drug that has been determined to target a unique genetic alteration. The use of cutting-edge screening technology will help identify which patient is a molecular match to each arm. This will create a rapidly evolving infrastructure that can simultaneously examine the safety and efficacy of multiple new drugs. We want to get the right drug to the right patient at the right time. This is good for patients because it allows them, with as many as 500 sites to be opened around the U.S., to have access to the drugs and allows us to study the effects so eventually they can become approved and be available to even more people around the world. What are the benefits of the LungMap? Enrollment efficiency. Grouping these studies under a single trial reduces the overall screen failure. That's great for patients. Operational efficiency. A single master protocol can be amended as needed as drugs enter and exit the study without having to stop and restart. Cost efficiency. As a result of shared services, utilization of existing infrastructure, and avoiding redundancy, this public-private partnership will operate at a cost substantially less than individual trials. There's consistency among trials, predictability in the outcome, full transparency with an oversight committee and a drug selection committee, benefit to patients, and seamless movement from phase 1 to 2 trial design. In fact, the FDA was very closely involved with the idea for this whole concept. My time is running short, but I'll tell you that I hope that this committee can help us with the issue of biomarkers, how to develop better biomarkers for these trials, how to regulate the diagnostics for these trials, Certainly the public-private partnership that we've developed is one that needs to be enhanced and helped and incentivized. And of course, finally, resources. We've been working with the NCI, and the budget is flat at best, and certainly we want to bring more of these drugs to patients. So as I conclude, LungMap is a public-private partnership where each sector has committed to do business differently. Together, we believe that LungMap can demonstrate a new model for high-quality drug development in less time, at less cost, for more people, and most importantly, improve the lives of patients with lung cancer. I'm happy to report the first patient on the study enrolled at Yale yesterday. The shared goal of accelerating the pace in which new drugs are developing is the driving force behind this partnership. We know that this committee shares that goal, and so we thank you for taking on this important 21st Century Cures initiative. Thank you.
Chair thanks the gentleman, and now recognizes Dr. Khosla, 5 minutes for an opening statement.
Good morning. My name is Sandeep Khosla. I'm a practicing endocrinologist and Dean for Clinical and Translational Science at the Mayo Clinic in Rochester, Minnesota. I'm also the principal investigator of the Mayo Clinic Clinical and Translational Science Award, or CTSA, from the National Center for Advancing Translational Sciences, NCATS, at NIH. I salute the 21st Century Cures initiative, and am pleased to share some thoughts on the opportunities and challenges we face in bringing new treatments to patients. Mayo Clinic has facilities in 6 states and provides care for more than 1 million people annually from all 50 states and 135 countries around the globe. In addition to clinical care, Mayo has a robust research program, including clinical trials. Over the years, Mayo has conducted pivotal clinical trials in many areas, including diabetes, osteoporosis, heart disease, and cancer. Mayo Clinic won a Nobel Prize in Physiology and Medicine in 1950 for the discovery of cortisone and its clinical applications. Conducting clinical trials is an extremely high priority for Mayo. With the congressional investment in NIH over the past several decades and the NIH-supported Human Genome Project, we are now in a truly exciting era where there are more possibilities for understanding diseases and developing new drugs and new treatments than ever before. With these opportunities, however, have come significant challenges. To address these challenges, NIH Director Collins created NCATS in December 2011 to catalyze the generation of innovative methods and technologies that will enhance the development, testing, and implementation of interventions that tangibly improve human health across a wide range of human diseases and conditions. As astutely recognized by this committee, the clinical trials process needs modernization. NCATS is seeking to do just that by funding CTSAs at 62 sites around the country, thus essentially creating a network of potential clinical trial sites. The vision is that high-priority clinical trials, funded either by NIH or by industry, could be run very efficiently through all or part of the 62-site network. While implementation is not easy, there are 3 changes that would facilitate the work of the NCATS Clinical Trials Network. One is institutional review board, or IRB reciprocity, between as many of the sites as possible. Because each institution has its own IRB, there are frequent and often lengthy delays in multi-center clinical trials as each IRB reviews and eventually approves a clinical trial protocol. Reciprocity between as many sites as possible would mean that once the IRB at the primary site approved the protocol, that approval would be accepted by the remaining sites. Second, there needs to be much greater interoperability of electronic health records. This could allow, for example, study investigators to rapidly search for study participants across all 62 CTSA sites. Third, for a national network of clinical trial sites to truly function efficiently, there needs to be greater harmonization of regulations. For example, an investigator today must contend with different regulatory requirements from the Office for Human Research Protections, the FDA, and the Office for Civil Rights, all within HHS. Further complexity is added by state laws that may go beyond the federal requirements. What can Congress do to help facilitate clinical trials at the national level? I have 4 suggestions. First, continue to support the efforts of NCATS and the CTSAs through ongoing and, if possible, enhanced funding. Second, help develop policies that encourage IRBs to have greater reciprocity with other institutions. Third, urge HHS to accelerate progress towards interoperability of electronic health records. Finally, develop policies for greater harmonization of regulations across federal agencies and across states. Responsibility for modernizing clinical trials falls also on the shoulders of individual academic medical centers. Here are 3 ideas academic medical centers could consider to modernize clinical trials. 1, work to shorten the time required for study initiation through more streamlined contract negotiation with industry and for IRB approval. 2, because disagreements over the use of biospecimens often cause considerable clinical trial delay, work to develop a simplified biospecimens policy that is broadly accepted across sites and companies. Third, develop better electronic capabilities to enhance recruitment, screening, enrollment, and tracking of study participants. In summary, the opportunities for bringing new treatments to patients have never been greater, yet significant challenges remain. Congress can help this effort by supporting discovery science, NCATS in the CTSA system, and by removing roadblocks in the clinical trials process. Together, government, the private sector, and academic medical centers must all step up and do all we can to rapidly deliver discoveries to the people who need them. Thank you for your opportunity to testify today.
Chair, thanks, gentlemen. And now recognize Ms. Stafford, 5 minutes for an opening statement.
Good morning, Chairman Pitts, Ranking Member Pallone.
Make sure your button's pressed. Yeah.
Okay.
Thank you.
Good morning, Chairman Pitts, Ranking Member Pallone, and members of the Health Subcommittee. Thank you for the opportunity to appear before you today. My name is Paula Brown Stafford. I am President of Clinical Development at Quintiles, the world's largest provider of biopharmaceutical development and commercialization services. We have more than 29,000 employees globally, including nearly 10,000 here in the U.S. We're engaged every day in helping bring better medicines to patients faster. To give you a sense of our scope, over the past 10 years, we've enrolled nearly 1 million patients in clinical trials at over 100,000 investigative sites like Yale, Mayo Clinic. Our experience and our role as a facilitator of the process gives us a unique vantage point on where the challenges and opportunities are in the drug development process. We all agree the development process is too expensive, in excess of $1 billion per NME, and takes too long. Generally, that's 7 to 10 years. And yes, patients are waiting. Modernizing clinical trials is critical if we are to meet the goals we share of delivering medicines faster, at less cost, to patients who need them. Quintiles works closely with our biopharma customers and the FDA to find better ways to design and execute studies to meet this goal. And we have had many collaborative successes to date, yet there is more to be done. My remarks will focus on 3 areas for further innovation and a number of recommendations where Congress can help accelerate meaningful improvements. First, with nearly 80% of total drug development time and costs spent on clinical trials, we must focus on patients, creating better ways to find the right patients for the right clinical trials. The bulk of time to conduct a clinical trial is spent in finding patients that meet the increasingly complex inclusion-exclusion criteria of trials today. Improving data collection and accessibility would facilitate more rapid identification of patients suitable for clinical trials. Without new approaches and better access to data, patient recruitment will become increasingly difficult, especially as we work to develop cures that are more targeted or personalized based on genomics. Second, there's much more room for improving the process of conducting clinical trials, reducing the timeline for each trial by eliminating redundancies and inefficiencies, particularly in what's known as the startup phase, where it can take up to 18 months just to get to a point where a study is open for patient enrollment. Also, standardization of clinical trials. The protocols, the data collection requirements would help to reduce repetitive activities that happen across trials. Among private sectors, the Clinical Data Interchange Standards Consortium, CDIS group I chaired from 2012 to 11, has recently even created data standards for a number of therapeutic areas, including multiple sclerosis, Alzheimer's, and asthma. The third area is pathways. Alternative development pathways could speed the introduction of new therapies to address serious unmet medical needs as an alternative to the traditional 3-phase clinical trial paradigm. Great strides have been made by the passage of FDASIA. The anniversary is today, 2 years ago today. Also, the creation of the Breakthrough Therapy designation and other expedited drug approval pathways. However, these have largely addressed FDA review time, which was 10 months, but not the much longer development time, which is 10 years. So how can Congress help? A number of recommendations. One, Congress could encourage the FDA to set goals for more frequent use of master protocols and adaptive designs. Both of these approaches allow multiple drugs to be evaluated in the same trial, identify affected and non-affected populations faster, and Quintiles has recently submitted a proposed master protocol for diabetes, CVOT, to the FDA and are expecting comments later this month. Congress could take steps to improve the quality and accessibility of data to researchers and thereby improve the speed and accuracy of identifying the right patients for the right trial. Among these steps are incremental improvements to linkages between EHR and clinical research databases, better interoperability among EHRs, and examining where there are misinterpretations of HIPAA and other data privacy regulations that may be inadvertently hampering the use of de-identified data to improve research. Congress should explore ways that the FDA and the NIH could encourage the use of central IRBs, which in our experience can cut the time, even start an individual investigative site from more than 100 to 45 days. And Congress could encourage FDA to pilot alternative development pathways similar to the adaptive licensing approach that the EMA is now piloting. The tools and science are in place to support alternatives whereby treatments could be tested and approved for limited use while ongoing studies would still be required. Chairman Pitts, members of the subcommittee, I ask you and your colleagues to support these recommendations because at the end of the day, a spouse, a family member, a friend, or even you may benefit from the next drug discovery that a modernized clinical trial system brings forth. Thank you.
Chair, thanks, gentlelady, and thanks all the witnesses for a very thoughtful testimony. And we'll begin questions and answers. At this point, let me ask for a unanimous consent request to submit for today's hearing record 4 items: letters to the editor of the New England Journal for Medicine questioning a number of assertions made in an article Dr. Kesselheim and others had published in the same publication on March 27th. And these letters include a letter from Mark McClellan of the Brookings Institution and Ellen Siegel of the Friends of Cancer Research, a letter from the Infectious Diseases Society of America, and a letter from the Leukemia and Lymphoma Society. Without objection, so ordered. I will now begin the questioning and recognize myself 5 minutes for that purpose, and I'll start with you, Dr. Siegel. Despite advances in science and technology, the duration, cost, and failure rates of clinical trial costs have grown exponentially, leading to delayed access and higher costs for patients. How can we reverse these trends?
Well, I think there's a number of topics that have been touched on today that could help address the issues around duration and costs and failure of clinical trials. Those would include the establishment of networks that can allow one to plug in, either through trials such as LungMap or through series of trials, new therapies into relatively standardized approaches with standardized startups and experienced investigators and standardized protocols. The better use of biomarkers and integrating them into trials, genomic and proteomic information to identify patient groups at risk, to identify early responders, and the use of those sorts of data to adapt trials while in conduct also offer the opportunity to reach either success or failure faster with a product and thereby to reduce the costs of product development.
How can we improve the process by which FDA qualifies novel drug development and review tools such as biomarkers and patient-reported outcome measures? And what would this mean for modernizing clinical trial designs?
Is that directed to you?
Yeah, Dr. Siegel.
It should be clear, first of all, that any sponsor, company, or investigator can propose for any trial the use of a patient-reported outcome or a biomarker, regardless of whether or not the FDA has qualified it. The qualification process allows broader use and acceptability and is intended for use with when many groups want to come together and bring together the data that demonstrate the utility of a biomarker or a tool for a particular purpose. It does appear that that process has been relatively scantily used. I think with the creation of more consortia and networks focused on diseases, there's an opportunity to use it more. I do not have expertise in how the process might be improved.
Okay, Mr. Murray. What part of the clinical research process consumes the most time for medical devices, and what are the major reasons device trials are moving overseas?
There's a couple reasons. As I mentioned during my testimony, early feasibility studies and getting to the point of actually having the device ready to start a pivotal study takes on average 6.5 years and $36 million. That's because there needs to be assessments done during the early phase. Medical devices are physical constructs and oftentimes can only be evaluated effectively in humans. So those early feasibility studies are extremely important. So streamlining that early feasibility process, IRB reviews, legal reviews for innovative new technologies can take very long, and having a process that's more consistent and more predictable in an environment where each site doesn't have unique and different requirements will help reduce the delays. Additionally, in today's environment, we have the situation where a lot of scientifically valid data is already available outside the U.S., and the opportunity to incorporate that data and use it for informed decisions in the U.S. could radically reduce the
to pursue
cost.
that a little bit, given the current reality, what can Congress do to help FDA accept the data collected outside the U.S. to ensure American patients are getting access to the American innovations sooner?
Well, one of the opportunities to look at rebalancing the pre- and post-market requirements, and if you look at reducing slightly the confidence interval in the pre-market perspective, for example, if the confidence interval in a trial were modestly reduced from 95%, say, to 90% in the pre-market phase, that could radically reduce by as much as half the size of the clinical trials required. And as long as there is appropriate controls and mechanisms in place to continue to monitor those patients post-market, that would encourage more products to be approved and could reduce the time to market.
All right, Ms. Stafford, how can real-world data enable us to learn more about the benefits and risks of a product, both in the clinical trial setting and once a product goes to market, and how can electronic health records and increased data sharing play a role in this regard?
One way that it can help in terms of using the EHR is actually in the feasibility of a trial and using the data that we have in the real world to help us design the best trial possible, and using that data up front to even help us identify and find the right patients for the trials based on prior experience with similar drugs or therapeutic areas. And real world is our ability to, it really goes into the master protocol or the adaptive design, and really bringing in data sooner and helping to make those decisions sooner based on the real world information that we have.
My time has expired. Chair recognizes the ranking member. Mr. Pallone, 5 minutes for questions.
Thank you, Mr. Chairman. I'd ask unanimous consent to enter into the record an article from the New England Journal of Medicine by Doctors Darrow, Aborn, and Kesselheim, and also a statement by Ms. Daggett.
Without objection, so ordered.
Thank you. I wanted to start with Dr. Kesselheim. Some of you have cited the need to use novel or alternative trial designs as a way to modernize the way clinical trials are conducted, and I want to learn more about one of these in particular, the use of surrogate endpoints. We've heard a lot about this recently, most notably with the situation surrounding two drugs, Avandia and Avastin. And these drugs were allowed on the market based on a surrogate endpoint through FDA's accelerated approval pathway. So I'd like to ask you, Dr. Kesselheim, to explain to us a bit more about what surrogate endpoints are, because I'm not sure I totally understand what they are, and how they are used in accelerated approvals. Specifically, what are the benefits of using surrogate endpoints? What are the drawbacks or concerns? And how has FDA relied upon surrogate endpoints appropriately, or have they relied on surrogate endpoints appropriately in your view?
Well, a surrogate endpoint is when we're testing a new drug, or a patient wants to take a new drug or get a medical device, they're most interested in extending their lives or improving their symptoms or other kinds of real clinical endpoints. A surrogate endpoint is an endpoint that is not one of those endpoints but might predict that endpoint ultimately. So in the case of a diabetes drug, instead of the drug showing that it improves lifespan or reduces cardiovascular events, it might change the hemoglobin A1C value, which is a biomarker and a surrogate endpoint that may predict ultimately down the line what happens. And the goal of using surrogate endpoints is try to shorten the span of clinical trials that are necessary to test a new product. The problem is when a clinical endpoint, when a surrogate endpoint isn't connected to the final clinical endpoint and then doesn't predict the final outcome of the drug. And if a drug is approved on the basis of the surrogate endpoint, then patients may experience bad outcomes even though their A1C is slightly improved, or in the case of the tuberculosis drug, even though their sputum is slightly cleared, more cleared of tuberculosis. So, surrogate endpoints, in order to be used as a basis for new drug approval, need to be validated by being linked clinically, and that's a very difficult and long process and can vary depending on the particular surrogate endpoint. So, just take statins, which is a cholesterol-lowering drug, and most people understand that most people agree now that lowering your LDL cholesterol is a surrogate endpoint towards ultimately lowering your cardiovascular risk. Unfortunately, there are some cholesterol-lowering drugs like statins that do a good job of that and then are connected to, where the surrogate endpoint does predict clinical outcomes. There are other cholesterol-lowering drugs like ezetimibe, which lowers your LDL, but then is not necessarily connected to improved health. And then there are other cholesterol drugs like torcetrapib, which was a drug that raised your HDL level, that again which was thought in a surrogate to act as a valid surrogate, but then ultimately did not end up demonstrating actual clinical effects.
But what about whether you think the FDA has relied upon these appropriately?
So I think that the FDA has a very difficult job and relies on surrogate endpoints in certain in certain limited circumstances where either A, the surrogate endpoint has been validated, or B, there's a great unmet clinical need, and that was, as in the case that you mentioned, the Avastin for metastatic breast cancer case, where everybody believes we need more therapies for metastatic breast cancer, and this appeared to be a good surrogate. Unfortunately, it later turned out that it wasn't, and it increased mortality of patients with breast cancer. And the problem is that at that stage, it was actually very difficult for the FDA to then withdraw the indication and now to try to change clinical practice away from using the product because the surrogate endpoint had sort of caught on in the marketplace.
It's difficult for the FDA to know when they're valuable or not, in other words.
Right.
Well, let me just ask one more to run out of time. Dr. Meyer, you noted that you would caution against shifting confirmatory efforts to the post-approval setting. Can you just expand upon that a little, and what is your view on how FDA has approached a reliance on surrogate endpoints?
Okay, so as far as the proposals to shift the regulatory decision-making more towards the end of Phase 2, relying on real-world data for efficacy, I don't think we are at a point with the science where we can rely on that. The kind of evidence what we want for assuring effectiveness of a drug at the present time, I think, can only come through well-conducted, generally randomized trials. I think the fact that half the drugs that fail from Phase 3 to approval fail for efficacy reasons is a good example that even at the end of Phase 2, where there's a lot of promise, that may not be confirmed by randomized controlled trials. As far as the FDA's reliance on surrogates, I think on the main, they do a reasonable job on it. I agree that they're in a tough position there, but I think for the most part, they're very judicious about it. And while they may not always get it right, I think the public health balance is such that you would want them to do well most of the time, and I think they do well most of the time.
Thank you. Thank you, Mr. Chairman.
Chair, thanks, gentlemen. Now recognize the Chair Emeritus of the committee, Mr. Barton. 5 minutes for questions.
Mr. Chairman, I've not been here I listened on TV, but I wasn't here in person, so I'm going to pass, but I appreciate your courtesy. I think this is a good panel, and I think the issues they're putting before your subcommittee are excellent. But I appreciate your courtesy.
Chair, thanks to the gentleman. Now recognize Dr. Burgess, Vice Chair of the Subcommittee, for 5 minutes.
Thank you, Mr. Chairman, and again, thanks to our witnesses for being here today. Mr. Chairman, before I get to questions, I just have another unanimous consent request. That yesterday's Wall Street Journal, the article by Peter Huber, they did a collection of articles about how things could change in this country to improve things. In addition to the tax code and two-parent families, here was an article by Peter Huber about unleashing molecular medicine, dealing with the very issue that we have before the committee today. I'd like to put that into the record.
Without objections, ordered.
Dr. Herbst, let me ask you a question, because you touched on it, but you didn't get much chance, and perhaps you could expound upon it a little bit. The use of the laboratory-developed tests, the regulating I think you put it, the regulating diagnostics for clinical trials.
Right, right. So this is a big challenge because right now, for genetic testing, there are 20,000 perhaps tests, that look at 4,000 conditions. There are many different tests. So how are we going to regulate and develop the right test to use? In the master protocol, what we've done is we're using a next-generation sequencing platform, which is allowing us to look at 250 different genes prior to the trial and then assort those patients to one arm of the trial. So that's an example of where we've designed the test in with a trial. Hopefully, the whole principle of regulation will then occur. We'll approve the drugs with the test. So that's the hope.
Now, with the FDA reauthorization that we did 2 years ago, and thank you, Ms. Stafford, for recognizing that achievement. Nobody else paid any attention to the fact that there was a bipartisan, bicameral work done by Congress in an election year that actually worked. So I appreciate the recognition. But when we did that allow for the type of flexibility that you are requiring for these laboratory-developed tests? Do you think, as you use this next generation sequencing, that you'll be able to get through the regulatory requirements that you need to?
I believe so. It's a challenge because this is a new paradigm to do a multiplexed series of tests and then use the data from that to put patients on trial. But the benefit we have in this large public-private partnership of the master protocol is we're working very closely with the FDA and with the branch that regulates these diagnostics and getting advice from them. We're working closely with our pharma partners, and we're working closely with the group that we've chosen to do the diagnostic test. So hopefully we're meeting all the requirements so that should this work and should a drug actually show efficacy, we can then get these tests approved. But I think it is important to look very carefully at what test is being done, the methods, the validity, the reproducibility of those tests, because there are so many different ways of testing for the same thing.
Correct. And that was actually one of the unanswered questions in FDASIA. So I would appreciate your feedback to this committee. If you find it's working well or not working well, we actually need to hear from you on that, because we never actually came and closed the loop on that and came to a conclusion. Dr. Siegel, let me ask you a question. I mean, your company, and this is perhaps a little bit off topic for you because you were primarily talking about drug approvals, but on the device side, Johnson & Johnson just achieved finally a FDA approval for a device called Sedasis that assisted in the administration of analgesia and anesthesia for people who are undergoing minor procedures. I don't know, minor is by definition is someone else's procedure, but undergoing procedures that are not open procedures. Can you speak a little bit to the difficult, because that was what, a 17, 18, 19-year-old regulatory process that this device required, and it seemed pretty simple and straightforward. Can you speak to that at all? Are we better now than we were the last 17 years?
I think that Sedasis is an excellent device and an important medical advance. It did raise important questions because, in a sense, it's replacing the use of anesthesiologists in some cases, or at least it had the potential to replace use of anesthesiologists with a technology-guided approach to delivering anesthesia and ensuring that the patient is safely monitored. And that, I think, raised a lot of safety questions with the FDA. It would be fair to, so I think the FDA had some legitimate concerns. I think it would be fair to say that there were times in the process where those could have been communicated better, handled a bit more expeditiously so that the process would not have drawn out as long as it did. But
well, the idea behind giving people a predictable pathway going through this process was largely because of the experience that your company had, and I hope FDASIA actually has dealt with that. time is short, but Ms. Stafford, let me ask you, you have it in your written testimony, you didn't get a chance to really get to it, but the sharing of pre-competitive data, how is that working out? How is that approach? Can you give us some real-world examples of how that works?
Thank you. It's a very good question. In terms of the pre-competitive data, it's having access to electronic health records so that we are able to take that data and de-identify it. We don't want to know who the patients are, but we want to know how to find the physicians who have those patients and enroll them. You know, the biggest time driver in this process, when we talk about these 7 to 10 years of development, is actually finding the patients. And when we talk about why do we go outside the U.S., it's partly to find the patients in a time frame in order to be able to get these products to market. And so the pre-competitive, if you will, data is really having access to data to help us find the right patients for the right trials in as rapid a time as possible. You know, right now, on average, you know, anywhere from 10 months to 4 years, and there have been trials that have been put together, and there's been some proposals put forward, and the ability to use data and to recruit the patients into a trial in 14 days. And just think about the amount of time that would be cut out of the trial from 4 years to finding patients and down to 14 days because we have the data that gives us access to identify the patients.
Mr. Chairman, I have additional questions, and I would ask unanimous consent to be able to submit those for the record. I'll yield back.
All right. Chair thanks, gentlemen. I recognize the gentlelady from California, Ms. Capps, 5 minutes for questions.
Thank you, Mr. Chairman. I thank you all for your testimony today. You know, providers and patients alike are relying on clinical trial data to assure that we're getting the right treatment at the right doses at the right time. However, for too long, these trials have not necessarily been representative of the population at large. And Dr. Kesselheim, I have a couple questions to ask you about this, but I wanted to just highlight where I'm going with my questions. Women have been excluded, assuming that women are, quote, men with hormones, quote. Even lab rats in the past have all been male, and recent past. And diverse ethnicities have been underrepresented. And even when these groups are included in trials, often there are too few participants in these groups to analyze the effects on them, or the analysis are simply not run or reported. More and more we are hearing about how disease manifestation can diverge based on gender. Recently, there was a 60-minute story examining how some drugs affect women and men differently. The story highlighted an example of the drug Ambien, which metabolizes differently in women than men. Because of this, women have been unsuspectingly receiving an inappropriately high dose of the drug for over 20 years. This FDA change was followed by a report entitled Sex-Specific Medical Research: Why Women's Health Can't Wait, which provides compelling evidence for the further inclusion of sex and gender in scientific research. And the FDA's own August 2013 report, which was initiated by the inclusion of My Heart for Women Act in the FDASIA legislation, show that there is still much work to be done to make sure that women are fully represented in clinical trial and that the safety and effectiveness of information is readily available. And to you now, Dr. Kesselheim, Brigham and Women's has been a leader in research on sex differences of disease. Can you tell us more specifically about the importance of ensuring proper analysis of drugs and devices on a diverse population? And what more can NIH, FDA, and private companies do to ensure that we don't have another Ambien situation?
Well, thank you very much for bringing that up. I think it's a really important point, and I think the essential issue that your question goes to is the generalizability of the study. And for a clinical trial for a newly approved drug or device to be truly generalizable, which is to say useful in the patients in which the drug will be used after approval, it needs to have representation of both sexes, people of different minority groups without relation to their financial status or their sexual orientation or any kinds of things. So the problem is that as we move in this conversation towards talking about more efficient trial designs and other kinds of processes to try to shrink the pre-market study, what that inherently does is it reduces the number of patients in which a drug is or device is tested in, and so makes it even harder to achieve the kinds of goals that you're talking about, and that have been recognized as being a problem in medical device trials of women underrepresented in trials of cardiovascular devices, or in trials of new drugs that will then be used in those patient populations. It's the same for older patients, and it's the same for younger patients. And so I think that the Congress, just as it can encourage the FDA to take up innovative clinical trial designs, can also encourage the FDA to make sure that the trials that are being delivered to it are fully representative of the patient population in which the drug or device will be used.
Great. And I want to get another topic in real quickly for you, because your written testimony also touches on the Sentinel system under development by the FDA to conduct post-market passive surveillance of drugs and devices to spot issues like adverse drug interactions quicker. And I believe that the Sentinel program holds great promise. It's why I work to get the Assurance for Effective Devices Act included in FDASIA to continue progress on the program and ensure it would be designed for drugs and devices. So can you discuss, there's only a little time left, how the Sentinel program could be complemented to the data derived from pre-market clinical trials?
Well, the Sentinel initiative, as you described, is a very promising pathway to try to get signals of safety issues for newly approved drugs and soon devices as well after they're approved. The problem is that the essential work in the Sentinel system of distinguishing the signal of the safety event from the noise of everything else that's going on with the drug in this post-approval observational setting is really very, very hard. And so in the last 6 or 7 years, the Sentinel initiative has been focused on the methods used to try to do this, and has made, again, relatively slow, steady, little progress, but steady progress in trying to assess these kinds of methods. I think there is still much, much more to be done before we can rely on the Sentinel initiative for any sort of real active surveillance, and I think that that's far in the future. But unfortunately, at this point, my understanding is that the funding of the Sentinel initiative going forward is still up in the air. So I would encourage Congress to continue to fund it, but I would also not get people's hopes up that the Sentinel system is going to provide this great white knight from a post-market surveillance point of view for drugs that are approved on the basis of limited pre-market study. I think the FDA itself still refers to the Sentinel initiative as the mini Sentinel pilot program now 6 or 7 years out from its creation.
Chair, thanks, gentlelady. Now recognize Dr. Murphy from Pennsylvania. 5 minutes for a question.
Thank you. I want to ask particularly about a couple of the issues related to psychiatric drugs. Certainly, many medications you've brought up with regard to some recommendations for advancing the speed of these are important. But in particular, with 60 million Americans affected in some level with psychiatric illness, 10 or 11 million with severe psychiatric illness, and about 3.6 million who are not in treatment in part because of not whatever the reason would be with medication, et cetera. Would there be some change in the recommendations you would make to advance or speed up research with regard to psychotropic drugs? And I'll open that question to anybody. Nobody has any there. Yes, go ahead.
Yeah, I will at least try to touch on that. I think I agree that it's an area of great unmet medical need. I think the problem has been a couple of fundamental issues. One is how poor some of the neuroscience is at predicting targets that are amenable to becoming drugs or targets for drugs. The second, though, is the fact that these trials are exceedingly difficult to conduct. And in fact, if one looks at drugs for antipsychotics and or depression, even very well conducted clinical trials often fail for effective drugs. So it's probably one of the more problematic areas to think about new paradigms of drug evaluation at the current time. I do think where the hope is for the future is really a better fundamental understanding of neurobiology to identify true opportunities for targets.
Let me add to that. Ms. Stafford, you also mentioned, I think, in your written testimony about issues involving, we should be looking at some of the EU standards, and perhaps that would help expedite. I know right now part of the discussion is also in terms of TTIP and looking at this transatlantic trade agreement, and those standards, I believe, should become part of that. Do you have any insights for us that you can provide with regard to some of the differences between the FDA, the American FDA, and EU standards for advancing clinical research?
Yes, I was specifically talking about the adaptive licensing pilot that was started in March, April of this year. So it's early stages in terms of Europe, and the FDA is having that discussion as well. So I don't think that they're too far behind, but I think encouragement to also pilot. There are a lot of different terms for this: progressive authorization, adaptive licensing, et cetera. And so that's the one major area that I was speaking to.
Thank you. I also have a question with regard to the HIPAA laws and how interpretation of those may interfere. I know some other members asked questions on this, but I also have some further comment to this, of how perhaps there are some barriers in what HIPAA laws are preventing us from getting information that would be extremely valuable in advancing research. I wonder if I'll open that up to anybody, if anybody would like to comment on changes. Dr. Sager.
Yeah, well, I think, since the time those laws were passed, we've had a lot of experience with them, and we have new types of information that can be collected in laboratories, and I think it is time for a relook. It's important that privacy be protected. I believe it can be done in ways that also facilitate the advancing of research, and I know that HHS actually had, about 3 or 4 years ago, an advance notice of public rulemaking that looked at both the IRB process for patient safety protection as well as the process for privacy protection. There's a lot of opportunity, I think, both to increase patient protections while at the same time allowing better availability of important medical information where there is minimal or no risk to patients.
Thank you. Dr. Herbst, do you have a comment on that?
Yeah, I just want to add one of the benefits of doing the genomics in the context of a clinical trial is then you actually have the informed consent from the patient, you're matching to the therapy, and then you have their consent to then do the discovery within the trial, hopefully identifying new targets for the future.
Do you think some of this is misinterpreted now by researchers or by physicians who are just afraid to go anywhere because of the HIPAA laws?
Well, I think people are concerned, appropriately so, and they follow them, and you do have to look very carefully at what consent you have whenever you're asking a question with tissue. But no, I think people are very aggressively trying to study what they can, reconsent patients when they can also, so that we can match genomic markers to activity.
Thank you, Dr. Khosla.
Yeah, I just wanted to add that particularly when you talk about clinical trial networks and consortia, I think that's where the HIPAA laws may need to be modified, particularly in what Ms. Stafford was referring to in terms of the kind of the pre-trial process. So before the subject has signed any consent forms, the electronic health record would need to be searched to identify participants at a given site. Currently, that data can't leave that particular medical center to be merged into data from other centers. So modifying that to allow that in a way that still protects patient privacy, but allows for better ascertainment of potential participants at different sites would be very helpful.
Thank you. So, the HIPAA laws as they stand, they were designed to help protect patients from exposure of confidentiality. They weren't designed to hamper research and other movements. I thank you very much. I yield back.
Chair, thanks, gentlemen. Now I recognize the gentleman from Texas, Mr. Green, 5 minutes for questions.
Thank you, Mr. Chairman and Ranking Member Pallone, and for our witnesses here today. In a time of historic opportunity offered with big data and scientific advances in technological development, it's important to examine the ecosystem of clinical trials. Before us is the prospect of transitioning from a reactive system centered on large patient populations, large clinical trials, and one-size-fits-all approach to a proactive system that can target smaller, specific patient populations, advance personalized medicine, and revolutionize the way we prevent, treat, and cure disease. Dr. Spiegel, clinical trial development in the area of antibiotics has been increasingly difficult in recent years because of the FDA trial design requirements. For instance, FDA requirements that trials study infection sites in the body versus the deadly pathogens that cause these infections that made conducting trials in the United States nearly impossible, in large part because of the small populations associated with these illnesses. How important it is to design to trial design successful trials. Is an FDA empowered to accept alternative trial requirements based upon the unique nature of the disease and the patient population? By the way, I'm sharing this question from Congressman Gingrey and I, who have legislation working on it. So is there something we can do that would make it easier on the smaller populations?
Well, clearly, infectious diseases are a major medical problem and threat to our country. Because of the rapid emergence of resistance and of new infections, and because industry efforts in this area have somewhat decreased, in part because of difficulties in pathways. But I think the issue before us is that the pathways that have traditionally been used and the way these drugs have been studied has been to develop them rather broadly for use, broad-spectrum antibiotics for use in large populations. And as your question presumes, what is needed is a better effort to focus on specific needs to develop drugs that can be used in specifically the populations that need them, so that resistance is less likely to emerge, and to have innovative pathways that will allow that to happen, and allow there to be ample incentives for investment in developing those therapies. I do think that there have been both legislative and regulatory moves in recent years towards in that direction, and I think that that's very welcome to in fact ensure that there are both incentives and pathways for more targeted treatments of critical infectious diseases.
Okay. Anyone else? Dr. Meyer?
Yes, thank you. I've actually worked on this issue, published on this issue, and actually I would say that FDA has shown some movement. I think one of the quandaries for FDA, however, is if they accept a smaller data set on a limited population for, say, a particular infectious agent, they don't really control the practice of medicine. And the issue for them is if they're reasonably assured that it works in that population, but they don't want it broadly used either because of poor antibiotic stewardship and or uncertainties about its general efficacy and safety, they don't have a good means for doing that. So I think that is part of the consideration that might be thought through in terms of approaching antibiotic drug development especially.
Well, and I agree in the real world of practicing medicine, but the FDA can put restrictions and advisories and things like that, so physicians may not, use that particular drug for things that may not be proven on the label. But I know that's, they don't have that ability in all the doctors' offices. So, Dr. Siegel, your testimony brings up the potential for continued recognition of surrogate endpoints by the FDA as having great promise for continued drug development in the United States. Over the past few hearings and roundtables, we've heard of a dire lack of new diagnostic tests for many of today's illnesses and conditions. As the adage goes, if you want to cure something, you first need to be able to identify what it is. Dr. Siegel, since such tests operate largely against predetermined endpoints, could early FDA recognition of diagnostic endpoints for the purpose of clinical trial design improve the efficiency and success of those clinical trials?
Yeah. First, I want to say, because on record, that the FDA program for accelerated approval has been a tremendous success. There's a large number of drugs, especially in cancer and HIV infection, that have come to patients much sooner, a large number of effective drugs that have come to patients sooner, and a large amount of increased investment in those areas. There have been cases, as has been pointed out, where subsequent studies have shown that those surrogate benefit endpoints did not predict benefit. That, in my mind, is evidence of the success of the program, the ability to learn in the post-marketing situation, and in fact, we have found, and when you just look at the numbers and the implications of the drugs involved, the benefits of those programs have tremendously outweighed the downsides, suggesting that more use, even though it would incorporate more risk, would be appropriate. Diagnostic tools are critical to do that. Diagnostics both to identify the right populations and, as you indicate, to measure endpoints. The use of diagnostics have been limited. The technological advances in proteomics and genomics and informatics offer the power to have explosive use. Dr. Herbst referred to some of that use in LungMap in all aspects of clinical trial designs. And I think that investment in research in that area and investment in ensuring that we know how to integrate in both the research process, the product development process, and the regulatory process, how we know how to integrate the development and the regulation of diagnostics with drug products is important, since historically they've been done by separate organizations or companies.
Mr. Chairman, I know I'm over time, and I appreciate it. This is such a great panel with so much information. If y'all have responses to not only my questions but other ones, please share them with us, and I thank you, Mr. Chairman.
Chair thanks the gentleman, and now recognize the gentleman from Illinois, Mr. Shimkus. 5 minutes for questions.
Thank you, Mr. Chairman, and I too appreciate you coming and been in and out, but actually been around in the little ante rooms and stuff. But I want to start with Dr. Koslaw. In your testimony, you state, and I'm just going to quote, the current clinical trial model for a placebo-controlled, randomized, double-blinded clinical trial may not be the most effective model, particularly for early phase studies. And then in the case of antibiotics, when you use I've been struggling with this, and I've actually been looking on my phone for the Hippocratic Oath and issues. So if you're using a double-blinded, placebo-controlled test, and you have someone in, I use the term emergent condition, and they are, maybe because it's in a test, you're using a placebo, doesn't that really cause ethical problems and challenges?
Yeah, I think you raise a very important point, which is the use of placebos in trials where effective medical therapy exists. And I should clarify that there have been enormous changes over the years in what is allowable and ethical to use as a placebo. So historically, for virtually all diseases, there were randomized controlled placebo trials. More and more, in my own area of expertise, for example, in osteoporosis, where we now have effective drugs to prevent or treat osteoporosis, instead of a placebo, often there is a standard of care drug that's used, and the burden of proof is to show non-inferiority or superiority to the current best treatment. So that's a great point that you raise, and it's in the context of where there may or may not be effective alternative therapies available.
Yeah, and I was going through this because one of the statements, this is a modernized version, I will prevent diseases whenever I can. Prevention is preferable to cure. I am to care adequately for the sick. And when we're in a system like that, obviously we're not. If it's placebo, Dr. Siegel.
Yeah, it's important to note that the use of placebo in a clinical trial doesn't mean that the patient is not receiving a treatment. For example, with a new cancer drug, if there's already 2 drugs being given, and a new drug is added, a new drug comes along, some patients may receive all 3, the others may receive the first 2, but also a placebo, so that there can be blinding as to which treatment, but they're still getting fully standard treatment. So placebos can be very important in research, but should not be equated with lack of treatment.
Seems like this started some comments. And so, Mr. Murray, please.
Yeah, so medical devices, it's a very important moral ethical question, and there are instances for breakthrough medical devices where there's not an existing therapy, and you do a surgical procedure, and then, especially with an active device that is not turned on, so the person is not receiving therapy, that, I think, adds to the conundrum, if you will, and I think it becomes a major challenge that's unique for medical devices, especially in those breakthrough areas where there is, say, treatment-resistant diseases with no other options.
So let me go back to Dr. Koslaw real quick. Do you, as far as in this process that we just discussed, any FDA reviews or reforms that you would suggest that be helpful in this process?
Well, I think it really comes on a case-by-case basis, depending on the particular disease being studied, because for certain diseases, there are effective cures, and you're really looking for a drug that might be better or have fewer side effects. And in that case, clearly the use of a placebo isn't warranted. In other instances, there really isn't a good alternative, and standard of care may involve You know, for example, just giving nutritional supplements like vitamin D or calcium, and in those instances, using a active drug against that standard of care is appropriate. So it's a major ethical issue. It's something, though, that's very specific to each disease entity and the alternates that are available.
Great, thanks. Let me, for my final minute, let me go to Dr. Siegel. And you talked about proteomics, if I pronounced that right, and molecular diagnostics and genomics sequencing. So what do you believe Congress needs to do to address and ensure that the potential for what I guess the terminology is precision medicine can be realized by both developers and clinicians?
Well, I think the potential to utilize those technologies in the development of precision medication is critical. I don't know that there's a specific legislative need to change the rules or the way drugs are developed. I think that we have what we need in that regard. I do know, however, as we've seen with breakthrough therapies, that congressional attention to an issue, highlighting an issue, congressional exhortations, congressional direction of how federal agencies invest and spend their money can have a big impact. And I think in those areas, certainly enabling FDA and NIH to help enable those technologies and those developments could be very important.
Thank you. And I know, Chairman, you probably have asked, and we'll mention that there will be an opening record for questions. There may be follow-up questions based upon your response. We would solicit, and then we would forward to you if you do that, Mr. Chairman. I appreciate it.
Yes, we'll have follow-up questions. Chair, thanks, gentlemen. Now recognize the gentlelady from Florida, Ms. Castor, 5 minutes for a question.
Thank you very much, Mr. Chairman and Mr. Pallone, and thanks to the panel for sharing your insights today. Dr. Meyer, I know you were formerly at the FDA and you've worked in industry, so I'd like to get your insights based on that experience on a couple of questions. We've heard a lot today about various ways that clinical trials can be modernized, everything from increased use of technologies like electronic health records to increased use of alternative trial designs like surrogate endpoints and adaptive trial designs. A lot of what has been mentioned, I would assume, is outside the purview of FDA. I imagine a lot goes on in the development of drugs and devices that doesn't and shouldn't involve FDA at all. I'd like to hear your view on that. Do we have the right balance for the modern era?
So I think some of what we've been hearing is outside the purview of FDA. For instance, the use of electronic health records for pre-competitive screening of patients and understanding who the patient populations might be. That really is pre-regulatory as well. I think the expansion of the use of surrogates is clearly within the FDA's purview. I think the difficulty there, though, is not with the FDA. It's really identifying biomarkers or other assays that will be validated to predict outcomes. And that's no easy task, and it sometimes takes a very, very long time. If you take, for instance, Alzheimer's disease, everybody would like to be able to do much smaller, much more focused trials. But to date, the biomarkers we have not predicted therapeutic benefit. So there's no choice but to do large, long trials. I think the other thing I would say is that the FDA does, I think, at times have some reluctance to accept things like patient-based assessment, electronic assessments. And I think that is something that they could be encouraged to do. I'm not sure it needs legislation, but for instance, if you're a pulmonary patient and you're able to have a very reliable home spirometer and measure your airflow every single day, that's a very rich data source. But if FDA insists that those patients go into the clinic and be assessed in the clinic, that's actually inhibitory to patient enrollment to some degree, but also I think it produces a more expensive and complex trial.
Mm-hmm. Mr. Murray, do you think that the current regulatory scheme is meeting the entrepreneurial spirit that's out there? And I'll give you a great example. In my hometown of Tampa, we have a fantastic new center called the Center for Advanced Medical Learning and Simulation by the University of South Florida. I was so proud of it. I took Mr. Pallone to visit. I know Mr. Bilirakis has been there, where we're bringing together the medical engineers, the academics, the folks that can work through the business cycle, have the 3D printers right there, so they take the device right to the 3D printer, right into, you know, a computer analysis of whether it works or not. Does this regulatory scheme currently, is that going to be acceptable for the advances in technology and devices?
Excellent question. The genesis of MDIC was the recognition primarily from Dr. Jeff Shuren at CDRH and the commissioner that medical device technology is advancing at a rate that we've never seen before. You see it in the consumer and the mobile and the social media side, but you're seeing that translate over to healthcare as well. And so there was a recognition that the tools, methods, and approaches used needed to evolve. And to do that, we're working collaboratively in the pre-competitive space. And you mentioned 3D printing. That's an example where you're going to see the realization of personalized medicine, where using computational modeling and simulation, people will be able to have tailored custom devices that are fit them and meet their needs specifically. And where we are going right now, and I think the opportunity and the need, and we talked about this in terms of HIPAA and data, but there's tremendous amount of data that's available out there in terms of patients post-approval of devices. And if you will, if you had the opportunity we have right now donor selections, if we had people that would be data donors instead of organ donors, and then they would allow their data to be used, I think we could improve by orders of magnitude the quality and richness of those models and simulations to even improve more on that technology that is going to realize personalized medicine advancements.
Thank you very much.
Chair, thanks, gentlelady. Now I recognize the gentleman from New Jersey, Mr. Lance. 5 minutes for questions.
Thank you very much, Chairman Pitts. In the various testimony of members of the panel, you have discussed the challenges in attempting to coordinate the work of multiple institutions before and during clinical trials. Varying regulations and protocols make it difficult, I think, for institutions to communicate one with another. If institutions that are attempting to coordinate have difficulty doing so, what about those that are not working together? And what methods are currently in place, if any, to reduce redundancies in clinical trials? And what steps would the panel recommend to ensure that we are not doubling up on research or making the same mistakes over and over again? Dr. Siegel, yes.
Yeah. Well, there's been a lot of advances recently in terms of transparency of research results and rapid publication, and there's been a lot of growth of consortia, Transcelerate Pharma as an industry consortia, other broader groups to enable better communication and cooperation. I think, as you've heard from several members of the panel, one area, though, of better shared learning and cooperation that we see already but could see more of are disease-specific clinical trial networks and trials such as LungMap or organizations which bring together broad expertise. And one of the nice things about some of the newer approaches to that is that there are organizations that are not just, say, academic centers coming together with perhaps government support, but are also incorporating patient and industry expertise and input to enable better addressing of some of the operational problems as well as the scientific problems that they need to face.
Thank you. Dr. Herbst?
Yeah, I would agree with that. And just sharing our experience for the LungMap trial, we're looking to accrue 1,000 patients a year. And this is throughout the United States, really focused at the community, places that normally don't have access to these types of trials. So it really requires using the National Clinical Trials Network, and that network uses a central IRB. We heard about that from the panel, so that this trial doesn't have to go through a different IRB at each site, which can take weeks in some cases. So that's very helpful. I agree with Dr. Siegel. The commitment and working with all the partners, the pharma partners especially. The National Clinical Trials Network is being supplemented by the public-private partnership that we're working with, and we need to all work together with the FDA as well, because this would all be a failed effort if at the end of the day these drugs and markers couldn't then go for an approval of the drug. And then I think one thing that we all have to also consider, we heard a little bit about surrogate endpoints, is quality of life and patient-reported outcomes and how we're going to build those into the trials and work with patient advocates and with those groups early on.
Thank you. Yes, yes, Doctor?
So I just wanted to reemphasize what I had mentioned in my testimony, which is that NIH is investing in these clinical and translational science awards across the nation, and so this is a pre-existing network where there are going to be best practices incorporated over time. There's hopefully going to be increasing IRB reciprocity. So many of the obstacles that we've heard about hopefully will be reduced or eliminated. And it isn't disease specific, so it would be open to any disease for which there's a trial ongoing.
Thank you. To the panel, is there something more we should be doing here on this committee and at the federal level to make sure that this occurs in the greatest way possible for the benefit of the better health of the American people? Yes, Dr. Herbst.
Well, just getting back to the whole idea of the public-private partnership, I think it's essential. It's only, in my opinion, that's really one of the reasons the lung map is working well. Any way the committee could work to incentivize that to move forward, the pre-competitive nature. The fact that we have 5 different companies deciding to put their hat into our trial versus doing the trial themselves. Now, I would hope that at the end of the day, they'll see that this is the only way to find these small populations of patients. But they are taking a risk, and ways to sort of incentivize, to promote, to give them credit for that, I think would be important.
Thank you. Thank you. Yes, sir.
Yeah. And again, on public-private partnerships, but in particular with our partnership, which includes NIH, CMS, FDA, the ability to have a flexible, collaborative environment in that pre-competitive space, it is oftentimes very structured. I think it's FACA, if you will, that becomes an important consideration. So we have to be able to foster and encourage these kinds of partnerships in that pre-competitive arena.
Thank you. Yes, sir.
So I would, another thing that I would add is I guess I'm a little bit less optimistic than Dr. Siegel is about where things stand right now in terms of data transparency and the ability to share clinical trial data. And I think that this committee and Congress can do a lot to try to encourage and put in place systems and structures to allow sharing of clinical trial data to try to prevent redundancy in testing of new drugs and to try to allow different groups to learn from data that is currently right now held as a trade secret by many companies.
Thank you. My time has expired, and this has been a very interesting and informative hearing. Thank you, Mr. Chairman.
Chair, thanks, gentlemen. Now I recognize the gentleman from Louisiana, Dr. Cassidy. 5 minutes for questions.
Mr. Siegel, Dr. Siegel, Dr. Kesselheim, the sharing of the data, that is proprietary data. So is the obstacles of sharing that the company releasing it? I'm just asking.
Obviously, you need to have some protection of proprietary information in order for innovation to occur, in order to have incentives for innovation. However, when clinical trial data get to the point where what is learned about that data could be used to protect the safety of patients, if it's a drug that's already approved or there are other...
I accept that, but just in terms of expediting other research, and I'm just intrigued, it sounds like a great idea, but would the companies agree to it? You follow what I'm saying? I'm not arguing either point, I'm just asking this.
Yeah, no, we've put in place, through an agreement with Yale, a third-party review that will enable much greater access to our clinical trial data where needed for important medical research and patient safety. And we believe that that's not incompatible at all with protecting innovation and allowing...
I think it was the Michael J. Fox Foundation that in order to receive their grant, you had to collaborate prior to peer-reviewed publication. Maybe I have that wrong, but nonetheless, it seems like a nice concept. Now, I don't know the practicality of NIH. Does NIH require that? I don't believe they do they? Anybody?
I'm not 100% sure. I mean, I think I would also support that, what Dr. Siegel has said about his company and its innovative relationship with Yale is actually, quite a good model for, other companies, but it's relatively rare at this point. I think that, the NIH, when it funds research, should be held to the same standard as when companies fund research as well. But when research on products that are available in the market is done on patients, there's really no reason why that research shouldn't be available for further study and for greater learning by everybody.
Dr. Herbst?
Yeah, I'll just add, Yale's an NCI Comprehensive Cancer Center, and I do know that the new regulations for recompeting those grants do require even more collaboration between centers. So hopefully through that we'll bring the pharma partners too.
Dr. Khosla, you and others mentioned having a centralized IRB, but that's already allowed. The Western IRB is the central IRB for many others. Now, would Mayo cede their, knowing how prestigious Mayo is, would they cede their IRB approval to a centralized Western IRB, for example?
So I think the answer to that is that is a culture change that's occurring at many academic medical.
So let me ask, so that is merely a culture change, is nothing regarding statute or regulation? Is that, I'm asking because there seems like there is a certain institutional pride that institutions do not wish to cede. That truly seems more of a culture issue than statute or regulation. Is that correct?
Correct.
Yeah. Believe me, I'm from that culture. I understand the hideboundness of it. Now, you also said something which I found intriguing. Dr. Herbst shook his head, yes, that if you are doing the screening with genetic markers, that material, that information has to remain domiciled with the institution. And yet Southwest Oncology Group, I'm just asking, you have multiple institutions. If one of them has certain biomarkers, they cannot share that with the centralized, whoever is overseeing the entire study framework. Whatever you learn cannot be shared with that centralized authority?
Actually, yes and no. So first of all, the patient gets their data, so that's very important. So we're making this screening available to patients where they might not have had it or afforded it. And then, of course, the excess tissue does get banked, through the cooperative group structure, but that's now part of the national system. And
now, is that statute or legislation, or does that require an act of Congress? Oh my gosh.
No, the groups have tissue banks, and the tissue goes in the tissue banks. And with petition, anyone, it's a public bank, can petition the SWOG at some point if they have a study and they want to use this tissue.
Now, I guess what Dr. Khosla, I think you said is that if you do biomarkers, those results remain at the institution and cannot be shared with others. Did I hear that correctly?
No, maybe you misheard me. No, this all goes centrally. In fact, the whole beauty of this is we are profiling at 500 different places with the same technique where it all goes to a central database.
That's what I thought.
And that's the beauty of it. The point I was trying to make is we have very broad consent on these patients, all very carefully through the IRB, so that we are both putting patients on the drugs that we know now will work, may or may not work, but we're also able to discover new targets. So for the next 4 or 5 drugs that will come into the lung map, we'll be able to be more informed in what we choose.
Yes, sir.
So just to clarify, what I was referring to was the preparatory to research phase. So before the patient's actually been enrolled in the study, to search the electronic health record, identify patients at a site, that information before that patient is signed a consent form can't leave that site.
Yeah, but that's okay because you're going to immediately, I used to do clinical research and I'd have 10 patients who I knew were interested in a trial and we would, we knew from looking at their study, it's just that they had not had the formal testing. I don't see that as an impediment so much as maybe, and I forget if we did this, if it's illegal, I didn't do it. But nonetheless, I would say, listen, I have 10 patients whom I think I can enroll as soon as we start. There would be some sort of signal, knowing that it didn't guarantee, but it suggested it might happen. Is that an impediment, or is that just a...
It's an impediment to the extent that when you have these national clinical trial networks, it's sort of an ongoing process to recruit both a site investigator and the study participants. And so if you know up front where the patients are, then you can seek out individual investigators at those sites. So in that sense, it is an impediment, yeah.
I yield back. Thank you for your generosity.
Chair, thanks, gentlemen. Now recognize the gentleman from Virginia, Mr. Griffith, 5 minutes for questions.
That's okay. Thank you.
Thank you, Mr. Chairman. Appreciate that. I'm going to pick up on that real quick. There's a company out of Richmond that I've been really excited about. It's not my district, but it's close enough. And it's the Health Diagnostic Laboratories, and what they do is they do all this stuff on your blood, looking mainly at heart disease and diabetes. And I'm sure they could add to their form a consent in advance, because what they're doing is tracking biomarkers and then giving counseling to the people that they've done the blood work on, obviously with the oversight of the physician, but they're giving counseling and trying to help folks avoid heart disease and diabetes, and a lot of times those biomarkers are overlapping. And it just seems to me that that might be a good place, because they've got folks all over the country that they FedEx in their blood samples to, and I call it, they've Henry Forded blood lab work, and it's really exciting stuff. And it just seems to me that might be something that you all could look at and find a way, particularly if they get consent from their patients in advance. You might be able to track some of the biomarkers that you're looking for or some of the other things that you all are looking for, that you then can get rid of that impediment that you were talking about by having a whole slew of folks automatically identified who may have already given advance consent, at least to be contacted. Yes?
I was going to say, I think the operative word is consent. And as several of us have discussed, it is a matter of designing your consent up front that allows you that capability. And, you know, for instance, we have a tool, a technology, Metagard.org, where we have about almost 3 million patients that we have data, we have a relationship with, but we consent them with them to participate in real-world research with us, et cetera. So I think it's about the consenting and what you put in that up front.
Absolutely. I would never want anybody's information being shared without their consent. What do you find in your getting the consent up front? What do you find? It was about 5 or 10% that say they don't want their data being passed along?
I don't have the metric, but it's interesting how many people want to be in the conversation, how many people are members of different, groups like the ADA, American Diabetes Association, or Multiple Sclerosis, and where they find their communities and how interested they are in research opportunities. And so our database is really, do you want us to communicate with you, because they're all very interested in being part of research.
And you all mentioned it earlier in your testimony today that the technology and things are moving so much faster than they used to move, and it's exciting, and it really has great opportunities. I'm going to switch gears a little bit, although it does connect. I think about these issues of developing new treatments, and I have to tell you, I align with the mindset of those who support right-to-try laws that are being passed in the states, and I've introduced similar bills, 2 such similar bills here. For patients whose doctors have exhausted current medical options and have been told that the end of life is nearing, my feeling is, why should the federal government interfere if the patient wishes to spend their own money on experimental treatment plans? I have this saying, if I'm dying anyway, why do I need to be protected by the FDA? Because death is near and all treatment options have been tried. That being said, I think the issue of benefit-risk framework should be brought forward in the earlier stages of a study of a new treatment by allowing an informed and responsible access to medications after the establishment of safety could allow for a faster translation of the science and technology from lab to clinic while ensuring safety, benefiting patients, and at the same time leveraging our nation's leadership and investment to advance science and technology. One of the bills I've introduced, the Patient Choice Act, does this by creating a provisional approval process after a drug safety has been established to allow patients to have access to new treatment while the efficacy is still being tested. This is similar to how things are moving in Europe. I think this makes sense. I think it makes sense to empower a patient, as we've been talking about today, particularly faced with the dilemma of a terminal disease, to help move the ball down the field in the area of medical science and medical knowledge by fighting to save their own life with experimental drugs if they choose to do so, and even if they fail, the satisfaction of knowing that they may have helped save someone else's life. So then the question comes, because I know that a number of you, particularly Dr. Meyer, are generally opposed to this kind of a concept, but when you're faced with the subset of that terminal patient and their doctors have indicated that the current medical options have been exhausted, how do you tell that person that they can't spend their own money to try something that may not work, but that might hold some promise? Dr. Meyer.
So I'd actually like to address that very point, because actually from my experience at the FDA, it's usually not the regulators who are standing in the way of that. It's actually more often the companies, and there's a couple of considerations around that. Often they cannot charge, and going through the mechanisms to charge are very arduous, and they have to prove what their investments have been. The other thing is that it ends up dirtying their data, if you will. So you mentioned the patient maybe having the altruistic view of, even if I don't benefit, maybe others will. But unless they're in a trial of some sort, and their data collected in a rigorous fashion, they may not, in fact, contribute meaningful data to the evaluation. So I very much am sympathetic to that view that those patients who have no other options and there's a promising drug out there should get access to it. But I think it really requires a thoughtful look at the ecosystem around that, if you will, and you know, what is the problem, what's the fix?
And Mr. Chairman, my time is up. I know Mr. Murray wants to respond as well, but I have to yield back at this point. But
go ahead, Mr. Murray.
Thank you. I just would say patient choice, we believe, is an important aspect, and also the consideration for devices in that discussion. And to the extent that there are methods and methodologies to streamline how a patient may pay for a procedure, because that is a difficult aspect in this, especially it's in a clinical trial, and how adverse data might be considered if it's not in a controlled environment.
Okay. Chair, thanks, gentlemen. Now recognize the gentlelady, Mrs. Elmers from North Carolina. 5 minutes for questions.
Thank you, Mr. Chairman, and thank you to our panel. Ms. Stafford, I have the great honor and opportunity to be representing North Carolina, and certainly your operation and organization there, the world headquarters right there in Durham. And I just have a couple questions for you. Again, obviously, our goal is to try to make the system work more efficiently so that we can get these very important drugs to market in a much quicker, efficient manner that's safe and, you know, for all of our constituents. And my understanding, as we've learned about the clinical trial path, that the sponsors who are collecting the data, they have to collect so many endpoints, I mean dozens of endpoints, and you know, to demonstrate that the drug is safe and that it works. My question to you, in your opinion, I mean, how much data do we need, and are we collecting too much data? Is the data we're collecting truly efficient, or are we collecting so much data that it's just over an abundance? And can we find a process to narrow that down if that's the case?
Thank you for your question. And of course, I'm wearing my North Carolina blue,
yes,
you know, just to say. Anyway, it's a very good question. And I'm a statistician by training, and I have seen in my almost 30 years in this industry now, we collect too much data. There is too much collected, and a lot of that comes from the multiple voices at the table. And I do think that having the conversation up front, and I think the FDA wants to work with us on this, with the industry, but there are a lot of key opinion leaders in the design of the trials, which includes many academic centers and scientists who have different opinions, and they want to prove that the drug is efficacious and safe, but they also want to explore what don't we know about the drug, what extra information can we get that's beyond really the investigation of that
scope. I mean,
product.
I think there again, I think what you're saying here is that what we need to do is narrow the scope so that we can come up with the information. And certainly more information is great, and that can be used in many ways after the fact, but I agree. I think, so would you say that upfront, straightforward, more transparency and focus on the actual goals that are trying to be met should be put forward initially?
Yeah, well, most panel members here talked about the trial design, and I think it all comes into the design and trying to focus the design, and as you say, the scope, and focus that scope, and not enter into too much interesting extraneous data, which ends up taking time to collect the data. Once you have that data, what
then
do
you have to do something with
data?
it. Right.
And it's just very costly. So trying to focus the scope of the trial design is my recommendation.
And there again, what we're faced with, or we're seeing more of the trend towards global clinical trials. And here for our committee, we're looking at ways that we want to show incentives so that some of those clinical trials can be here and kept in the United States. Can you make one or two suggestions on how we can achieve that goal so that we're doing more of those clinical trials, or we're kind of returning back to a process where we're doing them here in the United States?
I think we're having that discussion today in terms of ensuring that the U.S. is at the forefront of innovation around clinical trials, and that as long as we are the leader today in clinical research, we need to maintain that by being innovative and by modernizing the clinical trial, and being in a position to stay that leader. You know, drug development is no longer a one country, one continent, or one region, but we can certainly ensure that we keep our heritage as the clinical research leader by continuing this innovation discussion.
Thank you. And I saw some other nodding heads. Dr. Herbst, would you like to comment?
Yeah, I would agree. You know, I'm a medical oncologist. Many of us who work in cancer have very busy clinics. There's limited infrastructure, you know, flat or declining public money. You know, we're bringing some of the private money in, but really anything we can do to streamline the process, you know, the burden on the staff. You ask a few more questions. That means a coordinator or a nurse has to spend some time, you know, fewer, you know, rooms available. We want to put more patients on trial. Putting 5% of patients in this country on clinical trial is way too low.
Okay.
We have to do 20, 30. Actually, everyone should go on a trial in these incurable diseases, and to do that, we really need to be as efficient as possible.
And Dr. Koshla, do you agree with that? Yes. Thank you. Thank you,
Mr. Murray,
Mr. Chairman.
you wanted to add something?
I would just say briefly for medical devices, the just-in-case perspective of what's going to be required at panel for breakthrough devices and not knowing up front what a panel might ask. So bringing that part of the process forward would be very helpful, and also allowing for more flexibility in the early discovery. So when a new device comes out, you learn something, and allowing for adaptive trial designs that incorporate and don't necessarily poison the data for the overall trial.
Very good. Thank you very much, Mr. Chairman, for extending my time a little bit there.
Chair, thanks. Gentlelady, now recognize the gentleman from Florida, Mr. Bilirakis. 5 minutes for questions.
Thank you, Mr. Chairman. I appreciate it. Dr. Herbst, again, I want to thank the panel for their testimony today as well, and I appreciate you holding the hearing, Mr. Chairman. It's so very important. Dr. Herbst, I'm impressed with your multi-stakeholder partnership that resulted in the lung MAP program. Lung cancer has a 5-year survival rate of less than 20%. The work that NCI designated cancer centers do is tremendous as far as I'm concerned. In the Tampa area, we have the Moffitt Cancer Center, as you know, which is the only NCI designated cancer center in Florida. They have a partnership which has resulted in the Oncology Research Information Exchange Network, or ORIEN. This is the world's largest, in my understanding, it's the world's largest clinically annotated cancer tissue repositories and data for more than 100,000 patients who have consented to the donation for research. In your testimony, this is my question, in your testimony you mentioned the importance of partnerships to accelerate clinical trials, as well as the need to examine the incentives, structure, and process to facilitate data generation, sharing, and collaboration. Could you briefly elaborate on this and how this should be done, please? Can you elaborate?
Right, and I do compliment Tampa on their work. They were one of the leaders initially in doing this personalized medicine network and bringing it together. And we're basically doing the same thing. The lung map is really, it's a truly national effort. And as I mentioned, it came from an NCI panel and from work at the Friends Brookings meeting. And the thing that's very nice about it is we're working closely with the FDA, with the Foundation for the NIH, and others. We want to really bring these drugs and this testing throughout the nation to the community. So the idea basically is to pick and do profiling in one specific way at all the different centers within 10 days, you know, because patients can't wait. They have advanced disease. You're right. This is even worse than what you mentioned, because this is squamous cell lung cancer, mostly a smoker's lung cancer, where there really are no other therapies to offer these patients, the most advanced widespread disease. And then we're randomizing patients to either the standard of care or to one of these new drugs based on the molecular profile. And we have 5 different drugs. So the way this has worked has really been a good concept, something that the academic community, the clinician community around the country, and the drug companies and the private payers see as a very important way to move forward. And we've all worked together, and it's taken a large amount of collaboration, meetings. It really is a partnership. And I sit here now, but there are hundreds of people who have been involved in this process, and I'm very proud of how we've all worked together, and we're doing it for the patients. And the other thing that's very important is the advocacy community has been involved with this from the very beginning, and they've advised us on some of the issues regarding disclosure and forms and consent forms. So we've really worked, this is really focused on the patient and bringing more drugs to patients quicker. And I just want to add, the FDA has just been so supportive of this process. Of course, these trials all have to go through the standard phase 2, phase 3 criteria. In fact, they're very strict criteria, but we've had advice as we move along. How do you integrate the markers into the trial? So I would say this is something that has to be emulated, and other diseases are already working on this. There's a trial in colon cancer that's moving forward, breast cancer, and others as well.
Terrific. Very encouraging. Thank you, Doctor. Dr. Siegel, you raised the issue of providing greater voice for patients in clinical trials. You mentioned that the investigators are only objective outcome measures, the investigators, but not information from patients like how do they feel, how are they progressing. How could investigators and regulators use qualitative data when making decisions?
Well, thank you for that question. I think it's an important one. It's easier, I think, and that's probably why there's a history of using things that can objectively be measured in the lab or life or death. But beyond with the exception of life or death, usually what's most important is how a patient feels. The science, there is a science behind how to do that. If you're not careful about how you do that, you can introduce a lot of bias. You can use tools that misweigh and that don't really represent patient outcomes. So that has been part of the reluctance or maybe the slowness in incorporating patient-reported outcomes. But that said, I think we're at a place where they can and should be incorporated much more broadly in almost all areas of clinical research.
Thank you very much. Another question for you, Dr. Siegel. Can you explain in layman's terms what adaptive clinical trials are, how they are different from traditional clinical trials? How has FDA viewed adaptive trials? I believe they've released guidance just a few years ago. And have adaptive trials been used in Europe? And what lessons can we learn from Europe?
Sure.
I'm not sure if that's been covered because I had to step out, but if you could elaborate, I'd appreciate it.
Yeah, not in any depth. So more traditional trials, you design the trial and how you're going to conduct it and how you're going to analyze it up front. And then at the end, you unblind the data and you do your analyses. That offers the advantage of avoiding a lot of biases that can lead to inaccurate assessments of treatment effects. In adaptive trial designs, you learn as you move on. You use biomarkers or actual outcomes in patients if they're available fast enough to understand what are the more promising therapies, perhaps, maybe putting more patients onto those therapies, changing randomization, substituting, changing, or selecting among doses, or even changing entry criteria. You could change almost any part of a trial. A lot of scientific work has gone into how to utilize adaptive trials because if done wrong, there are opportunities to introduce bias, but they allow real-time learning from what's happening within a trial, and therefore they can be extremely powerful tools in drug development. The FDA has been out in a leadership position in terms of providing guidance as to how they could be used in the regulatory setting. There is, of course, some conservatism because of the scientific challenges, but it's a opportunity to accelerate our ability, as you've heard about from Dr. Herbst, to accelerate our ability to learn within clinical trials, and I think it's one that is very much underutilized.
Very good. Thank you. I yield back, Mr. Chairman.
Chair, thanks to gentlemen. That concludes the first round of questioning. This has been another exciting, informative, important hearing. A lot of members have follow-up questions, so we'll send those to you within 10 business days. I remind members they have 10 business days to submit questions for the record. I ask the witnesses to please respond to questions promptly. Members should submit their questions by the close of business on Wednesday, July 23rd. Without objection, subcommittees adjourned.